In the nerve fibers, the action potential has different phases. We start off at minus 90 millivolt, RMP, resting memory potential, and we have to reach the threshold of minus 60 millivolt. This is when the voltage-gated sodium channels start to open up. Anything below the threshold is called subthreshold. We have here an example. 1, 2, 3 excitatory signals adding up, and at 4, we have finally reached the threshold required to open up the sodium channels. So once they open up, sodium has a very large electrochemical force, so the permeability of the membrane now rises because the sodium channels are open. Sodium will go into the cells very rapidly. Then we have this depolarization phase. It stops at almost 35 millivolts because these channels are now closed. So we see again here, at subthreshold, channels are closed. At threshold, they open up and remain open until 35 millivolts, where they close again. Now at this point, the permeability of the membrane to potassium starts to rise again. We have here many different types of potassium channels, voltage-gated, like a domino. They open up one after the other, so we maintain a very high permeability to potassium until here. Remember, the equilibrium potential for potassium is minus 94 millivolt. So we see here, since the permeability of the membrane to potassium remains very high, all the way from here down here, we can reach a so-called hyperpolarization phase, where the membrane potential is almost equal to the equilibrium potential of potassium. Now let's see how this compares to the action potential in skeletal muscles. If you take a look at these two, they look identical, but there are some differences. On the left, this is the action potential in nerve fibers. It takes about 1 to 2 milliseconds. In skeletal muscles, about 5 milliseconds or longer. Second difference, the resting memory potential of skeletal muscles at more negative level. It's actually so negative that it can't get any more negative, which is why we usually don't have the hyperpolarization phase in skeletal muscles. So these are the differences, the major differences between the action potential patterns in skeletal muscles as compared to nerve fibers. Now we move on to the patterns of action potentials in smooth muscles. We have different patterns. Again, before we dive into details, please always remember the electrochemical forces for sodium, potassium, and calcium. We start out here at minus 50 millivolt, which is significantly higher than the resting memory potential in skeletal muscles and nerve fibers. In order to depolarize the smooth muscle cells, we have to reach this threshold of minus 40 millivolt, the blue dashed line. We have here slow waves that are generated. I will tell you how in the next slide. And these slow waves, when they add up and reach the value of minus 40 millivolt, they generate the action potentials, the spikes. Now, these action potentials, the depolarization phase of the action potential is mediated by calcium. Why? We have here an abundance of calcium channels, very few sodium channels. And since the calcium channels here in a voltage-gated fashion open up, and we have a very large electrochemical force for calcium, it will go into the cell, thereby depolarizing the membrane potential to minus 10 millivolt almost. Then we have the repolarization phase, which starts here. We are at minus 10 millivolt. Now, at this point, the membrane permeability to potassium rises. Potassium would like to reach its equilibrium potential, remember, minus 90 millivolt, so it starts to leave the cells. It is also believed that the activity of the potassium channels here during repolarization is dependent on the activity of the voltage-gated calcium channels. Okay. Now you see here the cells, the so-called interstitial cells of Cajal. These are the cells that are responsible for the slow wave generation. Okay? It remains debatable how they generate the slow waves. It is believed that this is mediated by calcium channels, voltage-gated calcium channels, and some other channels that depend on their activities. Anyway, what you need to know is that the slow waves are generated in a periodic fashion by the activity of Cajal cells. This is not the only pattern that we have for action potentials in smooth muscles. We also have this type. This type of action potential, as you can see here, is much longer. These cells, they require a much longer time for contraction. We have depolarization, and then we have about 200 milliseconds or even longer. Here we have calcium. Calcium has two roles here. One is to mediate the contraction of the smooth muscles. There can be no muscle contraction without high concentration of calcium. The second role is to maintain the action potential. Finally, we will talk about two different patterns of action potentials in the cardiac muscle. One in the SA node, the sinoatrial node, and the other in the working muscles. These are the pacemaker cells here. The SA node is here at the right atrium. This is where the heart activity starts off. It fires constantly, the SA node. Okay? It sends off action potentials, so it doesn't have a stable resting memory potential, and it fires at the rate on average of, say, 70 per minute. Okay. Then the contraction of the heart requires the activity of myocardial cells, so they have a different pattern of action potentials. Let's see how this works out. Here we have an action potential pattern in the SA node here. Remember, these are the pacemaker cells. They keep firing all the time. They have no stable resting memory potential. Start out at minus 60 millivolt. Here we have three different types of channels. IF stands for funny channels. Funny channels because they behave in a funny way. They get activated once the memory potential is hyperpolarized. They are also not selective. So we have sodium ions going into the cells through these funny channels and potassium ions leaving the cell through the same channels. However, we have more sodium going into the cell than potassium leaving. So we have a slight increase of memory potential by the activity of the funny channels. Then, short time later, we have T-type calcium channels, voltage gated. They open up, calcium will go into the cell. Why? Take a look again. The equilibrium potential of calcium is at 134 millivolt. Very high. So calcium will start to move into the cell through T-type calcium channels. Short time later, we have L-type calcium channels opening up. Calcium still has a very high electrochemical force driving it inward. Calcium goes through the L-type calcium channels into the cells until this phase here. Now, after we go above the threshold, we continue until we reach about 0 millivolt. At this point, the L-type calcium channels close. So calcium can no longer go into the cell, despite the still very high electrochemical force. At this point, the memory permeability to potassium rises. Potassium channels, like a domino effect, very many potassium channels, open up at different values of memory potentials, and then we arrive again at hyperpolarization phase, phase 4, and it starts all over again. Finally, the pattern of the action potential in myocardial cells. We have here a stable resting memory potential. There's an excitation going on here, which opens up the sodium channels. Sodium channels, once open, sodium will go into the cells. So we go all the way up to here, around plus 20 millivolt. Then we have a slight decrease in the memory potential, mediated by slight outward movement of potassium, but then we have a dominance of calcium channels and calcium activity. Phase 2. In this phase, 200 milliseconds or longer, this is when we need a high concentration of calcium. The heart contracts. Contraction requires high concentration of calcium. So calcium has to remain high. Free calcium concentration has to remain high. So calcium here has a dual role, like in smooth muscles. One is to mediate the contraction. Second is to maintain the action potential. Then contraction is completed, the muscle starts to relax. Repolarization is needed. This is mediated by outward movement of potassium. Again, like a domino effect, many different types of potassium channels, voltage gated, open up one after the other at different values until we arrive at phase 4 again. So we start all over again. You see, we have different patterns of action potentials.